Okay, so what it has is, a motor drive that turns this impeller, and as this impeller turns, this provides good mixing of the reactor. So this stirrer drive over here is the source of momentum transfer. You're basically transferring momentum from a drive to the reactor. And then at the bottom, you have a sparger, which is nothing but imagine a disc with holes in it through which air comes out. So these are air bubbles that come out and then kind of spread throughout the reactor. And this is what is responsible for the mass transfer part of it. Because as I pointed out last time, the bubbles, the oxygen goes from the bubbles into the liquid where the cells live and consume the oxygen. And then what's not shown over here is that typically you also have either a cooling coil immersed into the reactor or a jacket surrounding the reactor. Let me see if it picked up my bookmark. No, it didn't. So let me show it to you. These are also should be familiar to you from. Uh, so here are some examples of mechanisms for uh, you know so let me just point out in our department most of the reactors are of this type for some reason. These are called the jacketed ones. They are no longer considered the best. These are called jacketed vessels. Today I'm told that you can get away quite well with this system, which is nothing but cooling coils. But in our department this is the one we have. Almost all of them. I mean they have a lot of problems, but for some reason not clear to me they've chosen to buy these. Coming back to now, the uh, picture that I had. Uh, Go back. So the first question to ask is, why do you need all this? I mean, you know, I mean, uh, maybe you guys haven't done labs, but if you must have been shown this stuff. Um, if you do microbiology, most of the stuff is done with the shake flask, right? Which is a little sort of thing on top to maintain sterility. And all you do is you take this little shake flask. The volume varies from 100 ml to up to actually 5 liters. I mean, most of the work is done between 100 milliliters and 500 milliliters. So the volume I should put down is 100 to 500 milliliters. And you basically put this into what they call an incubator shaker. That is to say, a device. So this whole thing is put into a device. This is not to scale, but you get the basic idea. It's put into a device that is maintained at a constant temperature, and it kind of rolls around horizontally. It shakes, and as it kind of shakes, it's called elliptical motion. The liquid kind of moves around. You get the agitation, you get the constant temperature, and the oxygen comes, from this place into the liquid and provides oxygen to the cells. So the question is, why do you need this at all? You know, uh, there is no need. I mean, you could just everything that has been invented, pretty much, has been invented in a shake flask. So I mean, I'll take actually the classical example of penicillin, which sort of has become almost like a legend. I think you've all heard the story about Fleming, who was doing some experiments and discovered some fungus, and all those experiments were being done with shake flasks. But the question then arises: why did they ever feel the need to work with a bioreactor? So that's the first question to address, so that you kind of know that it's something worth doing. So, are there any thoughts on? Does anybody already know? Has this already been covered in your previous classes?
Not really, sir.
Okay, so let me just. So there are two broad reasons um, why you would want to do that. The first one is actually somewhat obvious. Um, um, let's see, where is it? So I think I've already shown you. Um, so the first question, uh, see figures in chapter 8 and 10 of Doran just to get a feel for it. Now I'm going to move on to the next question. Why can't we just use shake flasks? Because those were the things that were used to develop it. And I'll do it essentially by taking the case of penicillin. So in fact, actually, historically, so let's say we're dealing with question two. Historically, uh, you could say that the field of biochemical engineering began, uh, it actually literally began with penicillin, began with penicillin production. And I mean, the story here is that see, penicillin was discovered in the middle of World War II. So I don't have the exact date with me, but it would be some early 1940s. So early 1940s is when, or no, maybe even before that. I think it's actually before that. I should correct myself. So the discovery that he made was made essentially, as I pointed out, in a shake flask, and this thing sat forever. Part of it is that actually nobody understood. I mean, he was not, they say, not a very effective speaker, didn't communicate very well. Nobody could understand what he was saying. But sometime 10 years or so after he did his work, people picked up on it. And by World War II, it's not surprising that there was a dire need. I mean, soldiers were getting wounded and dying essentially by sepsis. I mean, they would have survived if you could control the infections that they were getting. So the work kind of picks up. Uh, but now they know that penicillin works. It's very, very effective, and they try it out on a couple of people, and they are able to prolong their life. But they need penicillin in very, very large quantities, and they are not able to produce it. Right? I mean, now that gets me to the question. So maybe I'll. So it's a very, very. So this first point, actually, this is actually the more subtle point. Historically, it arose essentially as a consequence of attempts. If you need to scale up, um, so what the hell happened to my eraser? Okay. So historically, the first is you need bioreactors for scale up. Now, why is that the case? So let me first see if you can figure out. So one way to answer this question is, and this is the way historically it happens. Why don't we just simply take? You did this thing in a shake flask, let's say at 5 milliliters, right? So supposing you did this experiment, this is what Fleming did. He made the thing in a shake flask. This shake flask was, let's say, 500 ml. Why could you simply not geometrically scale it up? Like, just make everything. So this is zero point 0.5 liters. And if I make every dimension of this shake flask 10 times, keeping shape the same, right? So basically, I'm making a gigantic shake flask 10 times bigger in each direction. I'm going to get essentially something 10 multiplied by 10 multiplied by 10, which is 1,000 times. So basically, I'm going to get a massive shake flask. which has the volume 500 liters. This is also one solution to the problem, right? I can't see on the face of it anything wrong with it. I did something here, it worked. Why don't I just do it here? So now let me ask you. So let's go through it. So let's start first with the concept of heat transfer, which is I think a little bit simple. So let's take the case of heat transfer. So it turns out that the little cells that are growing here, they are metabolizing, and just like the human body that produces heat when it metabolizes, so are these microbial cells. So there's heat being generated. This heat is actually being lost through the surface of the shake flask. Okay. Now it turns out that you have no problem when you perform this growth in a shake flask, but the problem becomes massive when you move to this situation, the scaled up, thousandfold increase in the volume, you start getting major heat transfer problems. Can anybody guess as to why that would be the case? I mean, just take a shot at it. This is not something very, very fancy. Well, let me take it a bit further and see if anybody. Um, Let me take it a bit further. So the first point to notice here is that the heat generation, right? The heat that is generated inside that flask is proportional to the volume of the flask. And that's because, the cells are living inside the volume. So the more volume you have, the more heat you generate. But the heat dissipation, the heat loss, the heat loss is proportional to, you can see the way I have drawn the picture. It is in some way proportional to the surface area of the flask. Now, can you tell me why you would face problems if you increase the size?
So
Sir,
the increase in volume is much more than the increase in surface area, like the incremental increase in volume.
But why?
So because like volume obviously is in like three dimensions, so scaling a quantity in every singular dimension scales the volume like to the power of three, but the surface area only to the power of two.
That's it. So basically, I have increased, I emphasized that I increased the size while keeping the shape the same. If you increase the size while keeping, so basically, what I'm saying here is if this diameter is D2 and this diameter is D1, geometric similarity means that, and this height is H1 and this height is H2. What I'm saying here is that H2 over H1 is equal to D2 over D1. I maintained the ratios of the lengths. I basically just increased the size. When that happens, everything is increasing in the same ratio, all the lengths. So that means the volume goes up as, whatever, this goes up as D cube, this goes up as D squared. So that means as I keep on increasing, the heat production is going up much faster than the surface area. There will come a point in time when this thing will fail. So, what is the moral of this? Well, the moral of this is that this implies that as size increases, you need you need to find ways to remove heat. Well, there's no choice. You just have to remove the heat, and that's where, if you go back, I showed you a picture showing heat being removed. Right? I mean, there was a jacket or there was a cooling coil. These were the pictures I showed to you from, I believe, the chapter on heat transfer. I think it's chapter 9 or 10, something like that. So that's the reason why you need to remove heat. Well, so when you talk about a bioreactor, that's the reason why you have cooling coils and stuff out there. That's one reason. The second reason, let's also take a look, is now oxygen transfer. Actually, this single problem, the oxygen transfer problem, was what brought the engineers into the business. Until then, the microbiologists were the people dominating penicillin production, but they just couldn't make it bigger. The real problem they faced was not heat transfer. The first problem they faced was this is an acute problem. This happens in every reactor. Now, let's get back to this with the same picture. Well, maybe I should draw another one. So now, I do the experiment in a shake flask. And as I just told you, the oxygen is coming from the top of the surface. This is kind of closed with a kind of a cotton ball with aluminum foil on it to so this is pulling in air. You know, air can diffuse through the cotton, come in. It comes into this headspace. Let's call this the headspace. From where, at the interface, it diffuses from the headspace into the liquid. This is where the cells are, and once it diffuses into the liquid, then the cells have access to the oxygen which they consume. This is the way you grow them in a shake flask. And keep in mind that this thing is rocking back and forth. So this keeps the liquid agitated. Now, again, let's say I increase the volume by a factor of 1000x. So this is my much bigger reactor. Why do you think there will be a problem if I scale up? What leads to the problem? Can you now let me see if you can argue your way through it. So this is a huge problem. You just go up and it won't work. This was the problem. The scale up did not work. This did not work in penicillin production. So this was the first attempt to make a biochemical at the large scale, and I mean, and the biologists failed. So then they brought in a bunch of chemical engineers. So this is around, I believe, I think it must be early mid 1940s. This was basically done. Uh, the companies involved were Pfizer and Schering-Plough, but the people involved in it were chemical engineers. I'm asking you now. This is why is this impossible? I've just shown you the picture. Can anybody take a shot at it?
Sir, so why is this a problem, scale up and transfer of oxygen?
I mean, I'm asking you. I've already given you everything you need.
So the concentration of oxygen would then be different at different points in the, it wouldn't be well mixed. Yeah, and it would be different. So some places will have absolutely no oxygen and some places will have a lot of it.
Well, I mean, you're talking about the mixing problem, right? That. So basically, what you're saying is that perhaps as you move to the large scale, the liquid is not well mixed. Is that correct?
Yes, sir. It is not well mixed, and some points in the liquid might not have any oxygen at all.
That's could be one reason. Although I'll get to that reason next. What I was looking for was an argument very similar to the one I've already made. Can you see a similar argument that tells you right away that a surface area to volume type of argument? When I go back to the figure. I mean, so O2 transfer, let's say O2 transfer, 1 second.
So I guess again here you can see like the oxygen initially diffuses through the surface area of like the liquid, and then it has to diffuse through the entire volume. But the like again the same a similar problem should arise that the surface area scales up only like
Okay,
squarely.
let's start with one step at a time. So you're saying that oxygen transfer to the cells, right? Or to the culture is proportional to
The surface area.
surface area of the interface,
Yeah.
right? Now, what about the oxygen consumption by the cells?
So that should be like the entirety of the volume that is
Yeah,
like
that's it, right? But the O2 consumption of the cells is proportional to the volume of the culture. Now, if you take these two quantities, the surface area and this volume, as you the ratio of these two quantities in this one and this one will be very, very different. I mean, number one, the small scale will give you a relatively high surface area to volume ratio. That surface area to volume ratio continuously decreases as you scale up. So even if everything is happening at the same rate, right? I mean, by rate, I mean if the reaction kinetics are the same in the small scale and the large scale, the large scale will collapse simply because the scale has increased in such a way that um, the supply cannot meet the demand. That's the bottom line. So once again, here, so it is clear the lesson you learn is you need to supply extra oxygen. I mean, that's just common sense. Well, that explains when I showed you the picture of the bioreactor. Let me go back to it. See, I pointed out this sparger. Every bioreactor typically has some mechanism for providing oxygen to the liquid. I mean, and this is one way of doing it. Typically, the mechanism is to blow some air with create some bubbles. These bubbles then, which carry pure air or pure oxygen inside them, the oxygen from within these bubbles then goes out into the surrounding liquid medium where the cells can capture it. So this is the second reason why uh, you need a bioreactor. So think of a bioreactor as something containing all the bells and whistles that you need because the thing fails. The Shake Plus fails. You simply can't make it bigger. Um, now, let me finally go to the third one. This one's a bit tricky. This is actually something that we will cover later on. And the third one is the one you already mentioned, which is third one is momentum transfer. So actually, this we will actually see numerically. Um, Maybe the simplest way to do it is, maybe it's easier to understand. I'm trying to see. Actually, it just turns out that the ability to keep a liquid well mixed is very, very easy if you have a small amount of liquid. Once you start going to 1,000 liters and 10,000 liters, it's just the power that is required to keep the liquid well mixed is just prohibitive. You're not able to provide it. So first of all, if you were to simply take, just imagine this, you will have to imagine mentally. I'm afraid I don't have a simple model for it. If you were to take a shake flask, this is 500 ml. This is 500 ml or 0.5 liters. This is, let's say, 500 liters. If you just mentally try to imagine, if I shake them both at the same RPM, you will get almost no mixing here. You get poor mixing in the so bottom line is that for same agitation, mixing is poor. at the large scale, at the large scale. So this actually is what is discussed at length in chapter 8. So I should just basically point out this is the chapter 8 of Duran on mixing. So maybe broadly speaking, I can summarize the rule for you here. This actually also answers the question, which people often ask. I mean, actually, this question is often asked of chemical engineers and biochemical. What do you do? I mean, what do you do as a chemical engineer? Clearly, you don't do any chemistry, right? The chemists do the chemistry. Well, this is the heart of engineering, and that is the following. I can summarize it in 2 lines. The first line is one, that the reaction rates are the same in the small and large scales. So supposing you have a first order reaction happening in a 100 milliliter vessel, then you move to something that is like say 1000 liters. The first order reaction in the 1 liter vessel is or a 100 milliliter vessel will remain a first order reaction when you go there. However, so these are the reaction rates. So stated differently, reaction rates are preserved no matter what the scale of the process. But the transfer processes, and that by that I mean processes involving heat, mass, and momentum transfer, these are not preserved on large scales. And by not preserved, I mean that it can change. I mean, things happen well on the small scale, they don't happen well on the large scale. So, this is the reason why the chemical engineering curriculum has not only reaction engineering, it also has transport phenomena. Because when things get bigger, transfer becomes the major issue. You can't get the oxygen in, you can't get the heat out, or you can't get the momentum in successfully unless and until you find some alternative ways to do it. So, anyway, that is actually a generalization of what I have already said. The main point is I've given you one reason. This is the first reason. Why do you need bioreactors for scale up? Well, the moment you increase scale, you need extra ways of providing oxygen, of removing heat, of providing momentum. That's where the bioreactor comes in. This is one reason. Now let me get to the second reason. So the second reason, I guess I should number things a little bit differently. This should be maybe 1 and 2. Now the second reason is to get good quality, data, right? Under invariant culture conditions. So what I mean by this is, I mean, just now let's just imagine a shake flask. This doesn't require experiments, but you can just sort of imagine. Supposing I do things in a shake flask. Let's say I grow some cells. I mean, let's be more precise and take the case of, let's say you grow yeast cells. So let's say you grow Saccharomyces. And let's say, so you have Saccharomyces cells in the little shake flask, and you grow them, let's say, on glucose. Now, if you carry out this experiment, I think the stuff is done kind of qualitatively in microbiology, but we'll flesh this out later on quantitatively. If you take a look at what's happening to the cells, you will typically find the growth curve kind of looks like this. I mean, so if you plot Um, we'll describe this in more detail, but some measure of the cell density, the concentration of the cells, the concentration of the cells will kind of on a semi-log plot. So this is log, let's say to the base e or ln e of let's say x. It'll kind of go up like this, and then it'll kind of flatten out and then go like that. Well, the reason for that is that initially during this part, their cells consume glucose. Well, while they are consuming glucose, if you take a look at the medium, you will find that this blue line. So this is cells, and this blue thing is actually ethanol. What is going on here? Man, not working out. Let's just do this. Oof. This is ethanol. So what's happening here is, and I should also actually put down this red line here is glucose. So what really happens is that the cells first consume glucose. While they are consuming glucose, they expel ethanol. And so you can see the blue line, the ethanol is accumulating in the medium. And then when the glucose gets over, then the cells start consuming the ethanol. And so you see a second burst of growth. So the main point here is that actually, as you can see, when you do things in a shake flask, right, things are constantly changing. Yeah, I mean, in a shake flask, um, so in a shake flask, the environment, and by the environment, I mean the environment of the cells, which is the surrounding medium, is always changing. Is always changing. Well, that means that it's really hard to keep things constant. I mean, you're not able to really follow what's going on. Actually, if you, in some instances, so for example, I would add, what would be changing, for example? Well, the the oxygen concentration in the liquid decreases continuously. Can you see why that would happen qualitatively? I mean, as these cells, which are inside the flask, as their population density grows, the oxygen concentration starts to decline. This doesn't again. This just requires physical thinking. Can you see why that would happen?
So to metabolize glucose, they would require oxygen.
Yeah, but that's fine. But I'm looking for like a quantitative answer, right? I mean, so really the idea is simple, right? The rate at which oxygen is coming in over here, this is basically constant. But as your cells grow, the number of cells is becoming more and more. So essentially, if your supply is the same, but your demand starts to go up, then the same oxygen has to be shared between a larger number of cells. No surprise that the oxygen concentration goes down. So that's sort of the basic argument here. Actually, so when you do experiments in a shake flask, you can never allow the cell density to go beyond the point. I mean, basically, you'll run out of oxygen. The cells will be oxygen deprived, and very soon the growth of the culture will come to a halt or become extremely slow. Another thing that changes is almost certainly the pH. Because I mean, in this case, I've shown you that the cells were excreting ethanol, but they could just as well. So I mean, you could even be accumulating, things like acids. I mean, certain cells secrete lactic acid. Some of them secrete fumaric acid. If that happens, the pH starts to change quite drastically inside the culture, and very soon it'll come to a halt. Now, that brings me to so pH can change drastically in a shake flask. So all of these means that you need ways of. I mean, if you try to get any data, you can't let anything happen. You can't let cells grow too much. You can't let too much product accumulate, because all these things will change things so drastically that it will affect the growth of the cells themselves. So if you want good data, now I didn't mention this, but if you go back to this picture here, um, in a bioreactor, you also have a vessel. Actually, not one, but you have two vessels. One contains acid and one contains base. And you also have inside the vessel a pH probe. This pH probe measures the pH, and if so, you actually have a pH probe, and you also have outside here a pH controller. So basically, the pH probe sends its input to the pH controller, which contains within it a set point, right? So you may set point at say 7 pH. If the pH diverges from 7, say for instance, if it drops below 7, then the controller will inject base into the reactor and make the pH back to 7. If the pH goes above 7, it'll inject base and bring it back to 7. So this is the way the bioreactor allows you to maintain constant conditions. In much the same way, the same principle, right? It's just instead of a DO pH probe, what you have is a DO probe. This also we'll discuss at length later on, but you have like a probe that actually monitors how much oxygen you have in the liquid phase. If this level drops, then you can set up a control in such a way that either the impeller starts to turn much faster or the gas flow rate increases such that your oxygen levels are brought back to your set point. So you can maintain constant DO, you can maintain constant pH. Of course, and as I mentioned to you, the temperature is of course always maintained constant. So the bioreactor is also a way, coming back to what I was saying. So as I said, to get good quality data, right, under invariant culture conditions, so you can maintain a constant environment. And this actually, one more thing which I can only explain when we go a little bit deeper, is that it's much easier to sort of vary things. We will understand this. So I'll just put this down as something we'll take a look. In particular, the continuous culture, or what is called in chemical engineering jargon the CSTR, the continuous stirred tank reactor, allows one to get very high quality data. Okay, so are these 2 points clear? Any questions about it so far before I get to the next topic? Any questions on what I have said so far? I actually need to hear
No,
yes,
sir.
no. No
So
problem.
nothing as of yet.
Okay, so now I'm going to move on to the next topic. Um, uh, the next topic was um, how do you describe um, the next topic was the third question I wanted to address was how do you describe the behavior of a bioreactor mathematically? Yeah. So give me a couple of minutes. I mean, let me just get myself a glass of water to drink. You can also take a couple of minutes break, and then we'll start this topic. So you can take a couple of minutes off. It's 10:17, 10:19 right now. I'll be back in by 10:21. Okay.
Okay, sir.
एक चाय दे देना यार. Okay, can I start?
Yes, sir.
Okay. Okay, so the third question. Where is it? The third question was how do you describe a bioreactor? So, I mean, the main thing I want to point out here is actually, in a way, separate the easy part from the hard part. That's so. This is actually how do you describe a bioreactor? I should add very clearly, mathematically. So the main thing is to actually separate. So my goal here is to actually to make it clear to separate uh, the uh, I should say let's say the advective or transport or again this is again I'm separating reaction from transport phenomena and actually oddly it turns out that even though your transport phenomena course may have seemed like a real challenge because it has a lot of mathematics in it, actually, to, to understand these systems, transport phenomena is the easy one. It's the reactions that are hard in a sense that I'll make precise now. So, or transport, or some people actually call it hydrodynamics. Hydrodynamic part. So these are all synonyms, all three, from the reaction part. Okay, and the reason is that actually hydrodynamics is almost like mindless. You just take a balance. You've been given the balance. You just take the balance. But the reaction part, you have to be creative. You have to pull things out of thin air, literally, based on there are no there are no rules. You build reactions by observing them. So, I mean, if you like uncertainty, then this is an interesting area to be in. If you don't like uncertainty, it can be intimidating. It depends upon your personality. So, well, the easiest way is to go back to something you've already done, but perhaps do it more carefully. Was your mass energy balances taught by Sunil Nath?
Yes, sir.
So I guess
Yes,
we'll
sir.
have to repeat. Yeah, so I guess everything has to be repeated. So so what I'll do is I'll basically take a mass balance in a system with flow, right? So that will show you where the flow terms are, what the reaction terms are, and then we'll first tackle the reaction part because the reaction part is the part that's really challenging. So consider. So we'll do it by example, and the example is something that you have done in chemical engineering, even though mass and energy would have been a disaster. You must have certainly done it in chemical engineering. They must have done a decent job of it. So here is I'm considering a continuous reactor because in a continuous reactor you have both hydrodynamics as well as reaction, and it's easy to see which one is which. So this is the flow in, this is the flow out, this is the culture, these are the cells. So what we have here is essentially sterile medium flowing into the reactor. Uh, Sterile medium meaning there are no, so this is sterile medium. That means there are no cells going in. You've been sterilized the medium to the point where all the cells have been killed. They come into a vessel which has cells in it, and then you're also drawing a stream off. And this is the effluent. This is the stuff effluent stream which has cells in it, right? This is the basics of a continuous. So this is a continuous culture. Now, I do not know. Did, was this balance covered in your class? Mass energy balances, or did you do nothing? Can somebody speak? Uh, am I audible?
Yes, sir, you are.
So, does this look like something you've already done?
So like the reactor part of it, at least we did in chemical reaction engineering, but like I don't recall doing mass and energy balances in this sort of a system.
So let me just start from the basics. So what happens in this reactor is you have, let's say, some flow rate F, right? Capital F. This represents the flow rate at which, so this has units of liters per minute. Okay. You have some medium, right? The salts and so on and so forth coming in. And let's say, oh, I should also mention this. Uh, you typically set up the medium in such a way, this I will explain later on, such that only one of the components of the medium influences the rates inside the reactor, right? So I don't know if this is covered. Any system that has multiple components in it, any of the components can affect the rates at which things happen in that system. But for the sake of simplicity, people will often maintain all but one of the components in a large excess, such a huge excess that they have no effect on the system at all. Okay, so for now, you take this as a kind of a something that I'm saying, but it'll become clearer as we go along. So typically, you have only one component of the medium that is going to influence things inside the reactor. That's typically given the name growth limiting substrate. And this SF is the feed concentration, is the concentration in the feed of this growth limiting component. Okay. Now, just keep in mind that this is valid for systems that have one growth limiting component. And I could easily set up the experiment in such a way that not one but two are growth limiting, you know. And actually, if you look at natural systems, say if you were to go into a lake or a pond, I mean, who knows what is limiting? Maybe five things could be limiting at the same time. It all depends upon how starved the microbes are for which components it could be. But in a lab, one typically makes sure that only one is limiting by artificially constructing the medium in such a way that the component of interest is limiting and all the others are in excess. So they will have no effect at all. Okay? So F is the feed concentration of the medium going into the reactor. S sub F is the feed concentration of the growth limiting component. This is the only one we'll worry about. The others are in such excess that we don't even care. Um, and let's say F is also the rate at which stuff is coming out. Okay. Now going inside the reactor, we have no cells. So if I denote XF as the concentration of the cells, this is equal to 0. There are no cells going in. Coming out, let's say S is the concentration of the growth limiting component coming out, and X is the concentration of the cells coming out. And I am assuming that this reactor is well mixed. If it is well mixed, then that means that the concentrations coming out are equal to the concentrations inside. Is that part understood from chemical engineering? What is the meaning of well mixed? Well
Yes, sir.
mixed means that the concentrations are the same everywhere inside the reactor, right? In every single point of the reactor, you have the same concentration, and the concentrations coming out are essentially equal to that unique concentration that describes the situation inside the reactor. Is that good so far? I'm just setting the tone.
Yes, sir.
Is that okay? So now we are ready to take, for instance, the balances. I mean, you can have many more things which I have not considered, but maybe I should. So let's also call P the product. Clearly, the product might be something that the cells produce when they grow inside the reactor. Well, we're considering a situation in which no product is coming into the reactor, but of course, it could very well come out of the reactor. Are you with me so far? Yeah?
Yes, sir.
Okay. Now, before I proceed further, I have to make a couple of definitions. The first definition is, this should be familiar to you, is that the growth rate of cells, this the rate at which the cells produce themselves, is directly proportional to the quantity of cells. So since we're dealing with asexually reproducing organisms, so the asexual reproduction is an important assumption here. The more cells you have, the faster they will grow. So basically, I'm saying that if X is the population density, right, then I think that using this notation will be confusing. So let me remove this. So I will simply say this is proportional to cell density. Okay. Another way of saying this is that this is equal to mu times X. So the more cells I have, the faster I grow. This is the proportionality constant. This proportionality constant is called the specific growth rate. And another way to say it is that this is just saying the same thing in another way. Another way to say this is that the specific growth rate is equal to the growth rate divided by the cell density. And this is something, another way. So, different people have different names for it. In the microbial literature, they call this specific growth rate. But if you go, for example, into the people who maintain the census. They call this per capita growth rate. You know. So now I should point out that this assumption is valid only for asexual organisms or asexually reproducing organisms. I mean, the reason is, roughly speaking, we won't have to deal with it, but I should just mention it in a very, very crude way. If you're talking about sexually reproducing organisms, then the outcome does not depend upon the population density. Obviously, it depends upon the population density of the males and the females, and typically you get a squared term because, let's say, X is the population density of the males, X is the population, Y is the population density of the females, and let's say it's interactions between X and Y that lead to growth, then it'll be proportional to the product of X and Y. And if X and Y are essentially equal, then it'll turn out to be X squared. So if that crude argument is okay with you, we can move forward, because we will not be dealing with sexually reproducing species. I mean, that's a different ballgame altogether. So for asexually reproducing species, the growth rate is simply proportional to X. The more people you have, the more cells you have, the faster will be the change. And the proportionality constant is called the specific or the per capita growth rate. Okay. Now, the second thing is, you also have to talk about the growth. So whenever I say substrate, I mean growth limiting substrate. So substrate consumption rate Okay. Now, here also, it should be intuitively obvious that the substrate consumption rate, this is in units of grams per liter, grams per hour, I should say, grams per hour per liter, right? So this is the speed with which you are consuming per liter of the reactor. This is also proportional intuitively to what the hell is going on? This is also intuitively proportional to X, the cell density. I mean, after all, the more cells you have, the more you will consume, right? Now, I will write this in the form RS times X, right? This RS is nothing but, this RS is called, just by analogy, here I called it a specific growth rate. This I will call the specific substrate consumption rate. So, stated differently, the specific substrate consumption rate, this is just rewriting the same thing in another form. The specific substrate consumption rate, which I have given the notation RS, is nothing but the total substrate consumption rate, which I use the notation QS. So this is called QS divided by X. So this is basically, notice the difference in the units. This has units of grams consumed per hour per liter of reactor, whereas RS has units of, this is RS, this has units of grams per gram of cells per hour. So this is in per liter, but this is per gram of cells. I mean, I write GDW because grams of cells is typically written as grams dry weight. because that's how they're measured. You basically take the cells and dry them. So this, by writing GDW, you are saying something about the way you measured the amount of cells you have. But the main point to notice is that the volumetric, this is also called the volumetric, or in general, the substrate consumption rate, is basically based on the volume of the reactor. The moment you divide it by the quantity of cells you have, you call it the specific, you put the prefix specific, and that refers to the rate of consumption of the substrate per gram of cells. Is that part clear so far? This is just basically playing around with definitions. We have not done anything
Yes,
yet.
sir, it's clear.
And the same thing can be done for the product formation rate. I mean, now I can move faster. QP, which has units of grams per hour per liter of reactor, is essentially equal to RP, which has units of grams per hour per gram dry weight of cells, multiplied by X, which is in grams dry weight per liter. So this is called the product formation rate on the basis of volume. This is called the specific product formation rate. Okay, so far so good. So now we start with the mass balance, and essentially let's take the balance. Balance is being taken over, we define the space in red. So this is the region, the thing that I have marked with the red. This is the region, this is my control volume, and I'm taking the balance over this. And the balance is as follows. So first I take what is the rate of change, and I ask myself, what do I have? So let's just start with cells. This is the mass balance for the cells. Well, the total amount of cells in that control volume, which is essentially the liquid volume inside the reactor, assuming that the reactor volume is total V and the cell density is X. So keep in mind, this is the reactor volume in liters. This is the cell density in grams. This is in grams dry weight per liter. So this is giving me the rate of change of all the cells that are inside my stirred tank reactor. That is equal to the rate at which cells are coming into the reactor, which is F times XF, which is in the feed, which I told you is equal to 0 because the feed is sterile, minus F times X, which is the rate at which the cells are going out of the reactor, minus, no, plus the rate at which cells are being generated inside the reactor. But the rate at which the cells are being generated inside the reactor is mu times X times V. And why is that the case? Well, I just showed you that the rate at which the cells grow per unit volume of the reactor is mu times X. See, mu has units of grams dry weight produced per hour per gram dry weight, multiplied by X, which has units of grams dry weight per liter. This cancels out and gives you how many cells you produce per hour per liter of the reactor. Well, I have to multiply this by the total volume to get the total rate per hour inside the reactor. So that's why I get a V here. Is that part clear?
Yes,
Total
sir.
silence. Can I hear yes or no?
Yes, sir.
Let me just see. Can I get a better feel for what's going on? I can already see that the number of people is steadily dropping. Is it clear what I did? Okay, I guess there is no other way of finding out. Okay, so now if this is the case and the reactor volume is constant, so I say if V is constant, then you get dx dt equals F over V, right? The first term is 0, so all you get is minus F over Vx plus mu times x. This F over V, this is commonly known as the dilution rate, so I should put it here. Maybe I should write it down like this. Minus d times x plus mu times x, where d is defined as F over V. So this is called in the literature, it is called the dilution rate. And the simple interpretation of it, this is in liters per hour, this is in liter. So you can see that this is basically expressing the flow rate, F, in units of reactor volume. So you can read the dilution rate as how many reactor volumes have flown through the reactor per hour, right? Because, so let's take a numerical example. If F is equal to 1 liter per hour, right? And V is equal to, sorry, so this is, F is, let's say, 1 liter per hour. And V is, let's say, 0.1 liters. Then basically F over V is going to give you 10 per hour. So F over V is essentially 10 reactor volumes per hour. Okay? And the other way to understand this parameter is it is just the opposite. It is the reciprocal of the mean residence time. So the chemical engineers like to work with a residence time, which is V over F. But the biochemical engineers like to work with dilution rate. Dilution rate meaning how much is the reactor getting, how fast is the reactor getting diluted out, which is essentially how many reactor volumes have flown through it per hour. So can you see, did you understand this equation so far? Is that equation clear? Was it covered in your class, this equation, in mass and energy balances?
No, sir.
No, not
No,
covered?
sir. No, sir.
No, sir.
Can I just ask an honest question? Was anything covered in mass and energy balances? Did you?
Sir, a few things.
So, okay. I think I have 5 more minutes. So let me cover one more balance and then point out the hydrodynamic and the kinetic parts. So this balance I just constructed was for the cells. Now you can similarly construct a balance for the substrate, and the product I'll let you do on your own. It's just a repeat of this. For the substrate, if I have to construct the balance, once again I'll take the rate of change of the total amount of substrate which is in my control volume being the liquid inside the reactor. That is S times V, because S was defined as the concentration of substrate in the reactor and V is the total volume of the reactor. Now here, the inlet term is F times SF, because I defined SF as the concentration of the growth limiting substrate. The rate at which it is going out is F times S. And the rate at which it is consumed is RS times V. Sorry, I have to correct this. Rs times X times V. Why is it this quantity? Well, I defined the substrate consumption rate as Rs times V, Rs times X. That has units of grams per hour per liter. I have to multiply that by liters in order to get the rate at which it is consumed per hour. So keep in mind on the left hand side, the units are grams per hour. You can easily check because S has units of grams per liter and V has units of liter. So S times V has units of grams. This is the total amount of substrate that you have in the reactor. DDT of that will give you grams per time, which is grams per hour. And on the right hand side, every quantity has to have the same units. You can check. The first one, liters per hour. The second one is grams per liter. Here we have the first quantity, grams per hour per gram dry weight multiplied by grams dry weight per liter multiplied by liter. You can see that essentially we end up with grams per hour for the third term as well. Now, once again, if volume is constant, this reduces to ds dt equals, again, divide through by V, and you'll end up with dsf minus d times s minus rs times x. So I'll let you do the product balance on your own, but these two balances will already allow us to distinguish between the kinetic and the hydrodynamic terms. Notice that, so I'll just say this is called the hydrodynamic or advective term. Hydrodynamic or advective term. This is your reaction term. Over here, these two are your hydrodynamic or advective terms. And this is your kinetic term, reaction term or kinetic term or reaction term. Now, these have different effects, which we will sort of flesh out later on. The main point here is that if I had no flow, if D was equal to 0, so basically a batch reactor, you can see that the advective terms disappear and I have pure reaction. And that is what we will start off this. So when I said in my first lecture that we will start with stoichiometry and kinetics, we're basically saying we don't want to deal with anything having to do with transport or flow. That's out of the picture. We're only going to worry about the kinetic term. So we're going to basically talk about reactions happening in a batch reactor. Yeah, not worrying about effects that emerge from stuff flowing in or out. So no continuous reactor, no semi-continuous or fed batch reactor, just good old plain well-mixed reactors. These will give you only pure kinetics. Now, what does kinetics mean? Because that's the starting point of the first chapter. Kinetics means telling you something about what is mu. We have not said anything about what is mu. What does mu depend on? What is RS? So far, we have said nothing about what is RS. What is, the product, even for products, you'll have to define something called product formation rate. Let's call that RP. What is RP? These things require what are called phenomenological models. We'll have to postulate them from scratch based on data. So that will be the starting point of the first section. Is that part clear? Any questions? I think it's already past time. So any other questions? None? So we'll call it a day then, and we'll pick up on it on Monday. That will start off with models for description of mu, RS, product formation rates, and then we move forward from there. Okay, so the only thing you have to do between now and Monday is try out the product balance on your own, right? Just similar to the substrate, write the product balance. I'll tell you the final answer. Check if you get it right. So the final answer is dP dt, this is the product balance, equals minus D times P plus Rp times X, where this is the specific product formation rate. Okay, so try this out on your own. Thank you very much. See you next time.
Thank you, sir.
Thank you, sir.